So our final speaker for this session is Bereketeab Haileselassie. He is an instructor in the Department of Pediatrics, Division of Critical Care, working in the Department of Chemical Systems Biology, and is the recipient of an MCHRI Pilot Early Career Award.
Right, thank you so much for the introduction. So for the next 10 minutes, I'll be discussing the role of DRP1 Phys1 mediated mitochondrial dysfunction in septic cardiomyopathy. As we all know, severe sepsis and septic shock continue to be a significant cause of morbidity and mortality in the pediatric population. Recent studies have shown an estimated prevalence of about 8% in the pediatric ICU, with a mortality of about 25%. Majority of these patients tend to have multi-organ system failure at the presentation of the ICU, and at least 1 out of 5 leave with one end organ failure at discharge. Out of the myriad of complications associated with severe sepsis and septic shock, septic cardiomyopathy, or the presence of left ventricular dysfunction in the setting of sepsis, has been linked with an increased morbidity and mortality in both pediatric and adult populations. While the current pathobiology is really not well understood, mitochondrial failure has been implicated in recent studies to be linked with end organ failure in severe sepsis and septic shock. Under physiologic conditions, mitochondria function is maintained through a continual process of mitochondrial pruning termed mitochondrial dynamics. This is mediated through mitochondrial fusion, which is what you're seeing in these photomicrographs, where you have merger of mitochondria and formation of network of mitochondria that are able to have more efficient cellular respiration and ATP production. And this process is mediated through mitophysins 1 and 2, as well as optic atrophy 1. On the converse is mitochondrial fission, or mitochondrial division, which is the segregation of damaged mitochondria and removal through a process of mitochondrial autophagy, or mitophagy. And this process is mediated through a key protein, which I'm going to focus on, termed dynamin-related protein 1. While physiologic levels of mitochondrial fission are essential for mitochondrial quality control and efficient cellular respiration, excessive mitochondrial fission has been associated with decreased ATP production, cellular oxidative stress, as well as cell death. In particular, recent studies have shown that dynamin-related protein 1 interaction with mitochondrial adapter Phys1, or Phys1, has been associated with excessive mitochondrial fragmentation in both ischemia reperfusion as well as neurodegenerative disorders. Furthermore, peptides that are rationally designed to inhibit the interaction between DRP1 and Phys1, for example, peptide P110, have shown therapeutic promise in abrogating excessive pathologic fusion, as well as damage in these two diseases. While the role of DRP1 Phys1 mediated mitochondrial fragmentation is somewhat understood in neurodegeneration, its role in sepsis-induced end organ failure is truly unknown. So one of the hypotheses of this project is whether or not DRP1 Phys1 mediated mitochondrial fission contributes to end organ failure in the setting cardiomyopathy in the setting of sepsis, and whether or not inhibition of this pathway is beneficial in rescuing cellular respiration, ATP production, and cardiac function in these models. So to first understand the role of mitochondrial dynamics in sepsis, we used a cell line model of sepsis to evaluate mitochondrial morphology and compare it to function. We used H9C2 cardiomyocytes treated with LPS. We serially evaluated mitochondrial morphology over the course of 72 hours and compared it to cellular respiration and ATP production on seahorse oxography, as well as mitochondrial integrity and oxidative stress using fluorometric assays, TMRM and MitoSox. Results from our experiments showed that there is a change in morphology that is seen under the septic condition in these cardiomyocytes, where we see a change from more elongated networked mitochondria, which you're seeing in these co-stained images, to a more punctate, round-like mitochondria, which is suggestive of a fragmented phenotype. These change in mitochondrial morphology is also associated with a drop in cellular respiration, as seen by a decrease in maximum oxygen consumption. a decrease in spare respiratory capacity on seahorse oxography, and also a decrease in ATP production at 24 hours. When we looked at oxidative stress, we saw an increase in mitochondrial-specific oxidative stress at 24 and 48 hours in these cells which were treated with LPS. Now, as previously stated, DRP1 is a key protein for mitochondrial fragmentation, and it requires to be activated through phosphorylation and localized to the mitochondria to mediate fragmentation. And when we looked at mitochondrial fragmentation in the setting, we saw, phosphorylation in the setting, we saw that there is an increase in DRP1 phosphorylation in our cell line model, an increase in mitochondrial localization of DRP1 under the septic condition. To next understand the role of DRP1 and Fis1 interaction, we used peptide P110 in this model. We incubated with LPS as well as LPS alone in control, and we evaluated same metrics at 24 hours following treatment. And results demonstrated that there is a decrease in mitochondrial-specific oxidative stress with in septic cardiomyocytes that were treated with peptide P110, as well as an increase in mitochondrial membrane integrity in these cells, and somewhat of a therapeutic benefit in maximum oxygen consumption and spare respiratory capacity under seahorse oxography. And finally, ATP production. Next, to understand its role in our animal model, we used peptide P110 in a biopsy, using biopsy mice in an LPS model of sepsis. We stratified the animals to control LPS and LPS plus P110, and we evaluated illness severity serially after treatment. We looked at, for illness severity, weight, temperature change, and mouse sepsis scores as a surrogate for illness severity in this setting. We also looked at echocardiographs serially at 24 and 48 hours and compared them to baseline. And we did a survival analysis for a subset of animals, and we did timed SACs for 24 and 48 hours for these animals. Results from this experiment showed that there is a similar phenotype of mitochondrial fragmentation in the cardiac tissue of septic mice, as seen here in these electron micrographs, where you see more round punctate mitochondria that are more disorganized. And also you see a significant decrease in mitochondrial architecture as seen below in comparison to the controls. On the converse, when we looked at the mice that were treated with peptide P110, we do see that there is preservation of the organized mitochondria and also preservation of mitochondrial architecture. When we looked at cardiac function, we focused on evaluating velocity time integral across the LV outflow tract as a surrogate of cardiac output. And we did see that there is a drop at 24 hours in VTI in the septic mice, which was preserved in the P110 treated animals. When we also looked at illness severity, we saw a decrease in cumulative mouse sepsis scores in the septic mice that were treated with peptide P110. And we did see a small survival benefit in these mice. So in summary, DRP1 Fis1 mediated mitochondrial fragmentation seems to be an important contributor to end organ dysfunction, in particular cardiomyopathy and sepsis. And inhibition of this pathway seems to have therapeutic benefit in mitochondrial oxidative stress as well as mitochondrial function in both our cell and animal models of sepsis. And its role needs to be further investigated in larger animal models as well as potential clinical trials. I want to thank my mentors, Dr. Muckley Rosen, Dr. Bernstein, Dr. Monak, and also the DMR laboratory. And I want to thank everybody for giving me an opportunity to speak.
Okay, I have a question. It looked like you gave the P110 3 hours after the LPS to the mice. Have you looked at later time points to determine the therapeutic window after the initial insult within which it has to be given to still have a beneficial effect?
Yeah, that's a very fair question. So we started with 3 hours simply because that was the time that we saw a change in phenotype and drop in temperature and increase in mouse sepsis scores. We have not done the experiments where we looked at how much longer we could wait to basically give the peptide. The fact that it is able to rescue mitochondrial fragmentation after an LPS insult is promising. But yeah, you're absolutely right. We don't know what time zero is for a septic patient. Time zero could be at home or a referral hospital. So that's a very important experiment that needs to be done.
Okay.
Thank you.
Thank you all.